Good evening, and welcome to Science and Food. Thank you all for coming. So I'm Amy Rowett, the professor in integrative biology and physiology here at UCLA, and it is my pleasure to present you with microbes from your food to your brain. So this is the second event in our fourth annual series of Science and Food springtime events. So as many of you know, this lecture series spawned from a course that I teach here at UCLA, also called Science and Food, which is a science course designed for non-science majors. 5-7, many of you students are out there tonight. Where are you? There you are. All right, excellent. So the goal of my course and with this lecture series is to take something that's not very intuitive to many of us, science, and explain that with something we all know and understand, which is food. So from the course spawned our nonprofit organization, Science and Food at UCLA, our mission is to promote knowledge of food through science and science through food. So you can learn more about our three main initiatives in education, undergraduate education, research, and outreach on our website, scienceandfood.org. So before we begin this evening's program, I wanted to highlight how each of your contributions are really essential and for our initiatives, including this lecture series. So this is where your ticket sales and donations and contributions go towards supporting these efforts. So I'd encourage you to consider making a tax-deductible donation, and you can visit our website, the Support Us page, for more information. So you can also support us by buying a stylish Science and Food tote bag, which will be available at the end of the lecture. So in addition to your support, I need to acknowledge the support of some key people in this whole program. Laura Kainsinger, I don't know if she's around, she's probably running all over the place because she is instrumental in making this whole event happen and coordinating everything. Dean Sork, the Dean of Life Sciences here at UCLA, as well as Barney Schlinger, the chair of our Department of Integrative Biology and Physiology, for their undying support for this lecture series from its very conception. Also, Wendy Slusser, the head of the UCLA Healthy Campus Initiative, and Louise Eno, as well as President Napolitano's Global Food Initiative that also supports food literacy events such as this. Yeah. Okay. So there's numerous volunteers who, if we could advance to the next slides, I won't be able to acknowledge individually, but have all been essential in making this whole evening program happen, as well as some of our supporters who provided funding, as well as some of the tasting samples that you're going to be having tonight, Mother-in-Law Kimchi and Health Aid Kombucha. So a big round of applause to all of our supporters. Thank you. So the overarching theme of this year's lecture series is the future of food. And this is an especially timely topic given President Janet Napolitano's Global Food Initiative that's across all of the University of California campuses. This initiative addresses one of the critical issues of our time, which is how do we sustainably and nutritiously feed the world's population, which is expected to reach 8 billion by 2025. So where does microbes come into it? Well, microbes is a fascinating topic to us at Science of Food, and is especially timely because A, fermented foods are delicious and scientifically fascinating. Plus, we were wondering why kombucha became so popular. B, we are on the brink of understanding so much more about microbes now thanks to advances in science and technology. For example, it's been a huge challenge to be able to culture microbial communities in the lab. But now with new methods, there's movement towards developing in vitro culture of these communities. And there's also the emergence of new cost-effective ways to sequence DNA so we can really pinpoint and label the different compositions of these communities in our guts, for example, and in the foods that we eat. So the ratio of microbial cells to human cells is purported to be 10-1, which also provides even more incentive to understand more about microbes. But interestingly, this estimate was made in 1972 by a microbiologist. But recently, just a few months ago, results from a group of Ron Milo at the Weizmann Institute in Israel challenged this number, saying that it's actually closer to 1-to-1, one microbial cell for one human cell. So it's really important to understand microbes and how they affect our health. So also in the culinary world, thanks to leaders and visionaries like Sandor Katz, who we're going to hear more from tonight, home cooks are being able to harness more of the delicious benefits of fermented foods, and also high-end chefs are exploring new frontiers of fermentation in their cuisines. So this brings us to today's event, which focuses on microbes from your food to your brain. So today we're going to hear from leaders in the culinary world and science who are pioneering investigations into how microbes affect the chemistry in our brain, explorations into the vast diversity of microbial communities in our food, such as cheese, and new and innovative methods for fermenting foods. So I'm now going to introduce and welcome this evening's speakers, beginning in alphabetical order with Rachel Dutton, who we're thrilled to have with us today. So Rachel is a professor at the University of California, San Diego in molecular biology. She did her PhD at Harvard Medical School, and she's pioneered the use of fermented foods as experimental systems to be able to understand microbial communities and how microbes interact with each other. She's the recipient of numerous awards, including 40 Under 40 by Cell magazine, and was recently named Kavli Fellow of the National Academy of Sciences. And you can also see Rachel on the latest episode of Cooked, the Netflix series with Michael Pollan. I can't wait to see that. And she's also been profiled in the New York Times as well. So you can read more about her there if you like as well. So Rachel, come on out. So it's so great to have you here. And Rachel and I have known each other for many years, back from our days at Harvard, so it's fun to see you again, too.
Thank you.
All right, so next we have Elaine Hsiao. She's a new professor here that's joined integrative biology and physiology. She's the Delogi Chair in Biological Sciences. She was also an undergraduate student here at UCLA, so she's alum and an inspiration to all of the undergrads here. So she obtained her PhD at Caltech, and her work explores microbiome interactions with the nervous system. Thus, the title, the reference to the brain in the title of the talk. So she's also been awarded numerous awards by, for example, the Sloan Foundation, 30 Under 30 in Science and Healthcare by Forbes, a National Geographic Emerging Explorer, as well as recently pointed to the White House Office of Science and Technology Microbiome Forum. So, Elaine, welcome. Thank you for coming. So exciting to highlight some of our work of one of our newest faculty members. So finally, we're very excited that Sandor Katz has come to join us. He's a fermentation guru, and many chefs and home cooks idolize him as a hero of microbes. So he's been termed a fermentation revivalist and has also been featured in the New York Times. You can see a video documentary of him to learn more about his work and his life. He's the author of many books, including the James Beard Award-winning The Art of Fermentation, an in-depth exploration of essential concepts and processes from around the world. And he leads workshops and consults on fermentation in various places. In fact, this weekend, he's leading a workshop on Saturday morning, and also is participating in Pickle Day, who knew, on Sunday at the Grand Central Market downtown, where you'll be able to take home your own jars of pickles. So Sandor's work has really inspired home cooks and professional chefs. For example, Michael Pollan is quoted as saying, "The book has inspired me to do things I've never done before." Katz writes about the transformative power of fermentation with such infectious enthusiasm that he makes you want to try things just to see what happens. So we hope that this evening's event will inspire all of you with more inspiration and enthusiasm to try your own fermented foods and to delve more into the fascinating science of microbes. So welcome, Sandor. And it's especially great it's especially great to have Sandra here because for years, many of the chefs who've come through our Science and Food lecture series have, they've raved about you and your work, so yes. So tonight's event is a panel discussion. As some of you are familiar with, we begin with short 5-minute introductions or so from each of our speakers so we learn about their perspective on microbes, and then we'll delve into some conversation. We welcome you to submit your ideas and topics for conversation. There's cards that should be passed around. You can write your questions on and they'll be hand-delivered to the stage. And you can also tweet your questions, and I'll try to stay abreast of those developments. So with that, let's begin. So Sandra, why don't you start by telling us a little bit more about your foray into fermented foods?
Sure. Well, so it's funny as we were preparing to do this, I received this questionnaire. There were two questionnaires. One was if you're a scientist, and the other is if you're a chef. And I actually don't fall into either of these categories. So I mean, I'm not a food professional at all. I mean, I'm someone who's always loved to cook. Loves to eat, and 20-some years ago when I moved from New York to Tennessee and started keeping a garden, I was such a naive city kid that it had not occurred to me that in a garden like all of your cabbage would be ready at the same time. So that first year that I was gardening and I had a row of cabbage, I decided to learn how to make sauerkraut, and I actually learned from the joy of cooking how to make sauerkraut. And I loved it, and then that just led me into other fermentation experimentation. I learned about yogurt and got some kefir grains, and I started making country wines, elderberry wine, blackberry wine, things like that. I started keeping a sourdough and baking bread with a sourdough, and that just kind of led me into an obsession. And it was a personal obsession that I shared with my friends. Until I got invited to teach a workshop. And the first time I got invited to teach a sauerkraut making workshop, it surprised me a little bit that so many people were projecting their fear of bacteria onto the process of fermenting cabbage. So, the main question I remember fielding that day was, how can I be sure that I have good bacteria growing on my sauerkraut and not dangerous bacteria that might make me sick, that might kill my children? And so, people, like I learned that people, I mean, all of us who were, born in the 20th century were, indoctrinated by the war on bacteria to be fearful of bacteria. And so these, really traditional, really time-tested, really, really safe processes, people were projecting this generalized anxiety of bacteria onto them. So I just got really interested in demystifying this process for people, and that sort of led me into writing some books about it and further teaching about it. And to any of you who are at all curious about experimenting with fermentation, I would really encourage you to do it without fear. According to the U.S. Department of Agriculture, the number of cases of illness and food poisoning stemming from fermented vegetables that have been documented around the world is exactly 0. There is no case history. There is no reason for anybody to sort of be anxious about it. And, fermentation is an ancient practice that's practiced in every part of the world. And it's considered, a strategy not only for preservation, but for safety, for making foods more delicious. And also for health. I mean, the folklore of traditions all around the world considers products of fermentation to be, foods that help people to maintain good health. And there's certainly no reason for, anyone in the 21st century who has any interest at all in experimenting with fermentation to do so. Now, the one other thing I would say is that, there have been a lot of I consider fermented foods as a broad group, live culture foods, the fermented foods that are not heat processed or cooked after their fermentation, to be supportive of good health in a general way. There's certainly a lot of unscrupulous marketing where people are claiming that, eating a particular food will, cure a particular disease. And I don't really think we have the sort of, evidence in that way. But, foods that will potentially improve your overall digestion, your overall immune function, possibly impact upon your mental health in positive ways. I mean, that's very beneficial, even if, we can't really claim that it, cures diabetes or reverses cancer or cures any other particular disease. So anyway, what I do is sort of, teaching practical skills, demystifying the process, and helping make it accessible to people.
And so Rachel sort of bridges the gap in that you are working with people like Sandor and other cheese people and cheese. Cheese makers.
People made of cheese.
Yeah.
Yeah, so my research is
And there's some slides that should be projected.
If so, if not, it's okay. So my work is really trying to study these traditional fermentation processes, partly out of an interest in wanting to understand the fascinating microbiology that's there and how that impacts the food, but also my main goal is to try and use these fermented foods as simple ecosystems. So if we, think about the very complex microbial ecosystems that you find in pretty much every environment. So if you've heard about the gut microbiome, for example, which we'll hear more about, you have thousands of species living together, or every gram of soil, thousands of species. And so as microbiologists, we really want to understand how these microbial communities form, how they function, how the species that are living there interact with each other. But because many of these communities are so complex and difficult to work with in the lab, it can become really challenging task to try and sort out who's doing what and how they're doing it. And so I'm approaching this challenge of understanding microbial communities by using fermented foods as simple ecosystems. So these foods are very reproducible. So as Sandor said, you can make many different types of fermented foods, and you can pretty much rely on them to produce the same sort of product. And that's because the microbes involved in these fermentation processes form these simple, very robust ecosystems. So this is one of my favorite, or probably the favorite ecosystem of fermented foods, which is cheese. And so I really think of these as edible ecosystems. And what you're looking at here are actually microbial communities. So microbes are important for every step of the cheese-making process, whether it's the initial fermentation of the milk or the aging process. And these are all pictures of cheeses that have actually aged in caves. And during that aging process, they form these wonderful microbial communities on the surface, which we call the rind. So maybe next slide. And so this rind of the cheese grows on the surface of the cheese during the aging process in the cave. And it's a collection of bacteria and fungi living together. Next slide. And this is actually a very high-resolution picture. So if you imagine that you shrink yourself down to the size of a bacterium, which is really, really small, and you happen to find yourself on the surface of a piece of cheese, one of these cave-aged cheeses, and you look around you, this is what you would see. Okay, so this is a very densely packed collection of bacteria and fungi. So bacteria are these very small cells that we've been taught to be afraid of, but are critical for pretty much every fermentation. And fungi, in this case, the form of yeast or molds, the fungi are actually more closely related to us than they are to bacteria. So even though they're around the same size as the bacteria, the fungi are like our cousins, and the bacteria are sort of very distant cousins from both us and the fungi. So we're much closer on the family tree. And these fermented foods, during this process of fermentation, you get the growth of these very rich microbial populations. And so any fermented food that you eat that hasn't been processed or heated after fermentation will probably, every bite you eat will be millions and billions of cells that are alive. And so in a gram of cheese rind, this is from work in my lab, we estimate that there's about 10 billion living cells in every gram, so sort of like a teaspoon of cheese rind. And inside the cheese, you have a sort of a similar number of living cells. Next slide. And these, I just thought I'd show you some pretty pictures of some of these microbes. So just to kind of additionally show you that they're not scary, they're actually wonderful and beautiful. And so these are bacteria and fungi from a cheese rind, and the sort of round, orangish, and sort of star-like white colonies. We call these colonies. They're populations of microbes. Those are bacteria, and the fuzzy things, those are the molds. So next slide. And depending on which cheese you take them from, you have different communities. And so one of the things that we're interested in my lab is how diverse these communities are and how much it varies from different fermented food to another. So next slide. So they're just amazing how many different types of things we find. And next slide. Okay, and so what we try and do in the lab is we're taking these very traditional fermented foods and bringing them into the lab and trying to simplify them so that we can study them and understand how these microbial communities form, how they interact with each other, and how they're communicating. And so we try and mimic what the traditional fermenters, the cheesemakers, are doing. So in traditional cheesemaking, you make a cheese, you put it in a cave. I'm not sure why anybody ever thought that was a good idea, but it turns out to be a great idea because you get the growth of these microbes, and it's because of these cool human environments, and you end up with this sort of diversity of different cheeses because of these microbial communities. And so in the lab, we try and mimic these traditional processes. We take cheese and we sterilize it so it starts out very homogeneous, and then we pull the different microbes that we're interested in and mix them all together and grow them in cave-like conditions. And this is a picture here of one of our petri dishes. So it's actually about this big in real life. And each of those little wells is a sterile cheese that we inoculated with a different collection of microbes. And we're studying in these wells how the microbes interact with each other. Next slide, please. Okay, so that's pretty much the summary from my lab. And that's, our main focus is really trying to build on these traditional foods and use them in a very modern, scientific way and trying to understand how microbes interact with each other.
So next we'll hear from Elaine, who's studying microbial communities and how they affect our health in a different way.
Yeah, so this is the start of my slides. And so you've heard a bit about how we encounter a lot of microbes through ingesting them in these wonderful foods. But actually, microbes, many would argue that we live in a microbial world. So could you please click through? And so microbes are really integrated into a lot of processes, basic processes that make life habitable here on Earth. And so we've heard on the right here that microbes are involved in food and agriculture, but microbes are really rooted in nutrient production and in nutrient cycling, and also in industrial applications and in health and disease. And so aside from ingesting microbes and aside from this idea that we live in a microbial world, there's this recent appreciation that in many ways, each of us is our own microbial world. And so if you take a look at these pictures here, these are real scanning electron microscopy images from different body sites from a healthy human. And on the top left, this is an image at 3,000 times magnification into a human intestine. And what you can see is that it's jam-packed with lots and lots of bacteria and other microorganisms that are resident. So these are not pathogens or disease-causing bacteria, but these are normal bacteria and viruses and other microorganisms that are indigenous to our own human bodies. And if you look at these other body sites, so virtually every exposed body site, the mouth here on the top right, the stomach here on the bottom left, and even the belly button on the bottom right. It's really inhabited by several different species and types of microorganisms. And so there's a lot of recent interest and advancements in technologies to be able to even profile, out of all of these millions of bacteria, what are their identities and what might they be doing. And in the laboratory, there are many groups that are interested in learning how they affect a variety of aspects of health and disease. So next slide, please. And so if you just click through, please, in the slides, there are a lot of different statistics that have come out by very large efforts, federal efforts or national efforts to learn more about the microbiome. And I should emphasize, this is a really new area of research because only in the past 5 to 10 years have technologies developed and advanced, mainly sequencing technologies, to even profile the microbiome, to catalog what microbes are there. And so from these types of studies, you heard in the introduction that it's estimated that just counting bacteria, not viruses or other microorganisms, that bacteria in the human microbiome are at least 1.3 times up to 10 times more abundant than our own human eukaryotic cells. And actually this range, this discrepancy depends on whether you count red blood cells. So red blood cells don't have a nucleus. So if you count them, then you would get this bottom range of 1.3, and if you don't count them, then you would get 10 times more bacteria than our own human eukaryotic cells. And there are a lot of different statistics about how microbes contribute many, many more genes in absolute number compared to our own human genomes. And even this really neat estimate that if you take all the microbes that live in and on the human body and pack them together, that they might weigh the size of a pretty hefty organ, so about up to 6 pounds. And so there's this recent appreciation that the microbiome can have really important roles in health and disease and normal functioning of the human body, and that it was previously understudied. So many people consider it a forgotten organ. In terms of recent developments and what the microbiome can do, there are a lot of different implications. So for one, this was known for quite a while, but the idea that microbes, especially in the gut, influence digestion and energy metabolism. And so this partnership between humans, ourselves, or mammals, and the microbes is so strong that there are certain dietary components, like complex polysaccharides, that we depend completely on microbes to help metabolize for us. And in recent studies, there are also labs across the world that have identified certain bacteria that modulate the immune system. And so these certain species can influence whether the body responds in a more inflammatory way or in a more immunosuppressive way. And there's really intriguing studies about how the gut microbiome, so microbes that live in the intestine, can influence our responses to normal medications. Things as prevalent and common as Tylenol can be differently metabolized by different types of bacteria, and these changes in the microbiome might underlie how I metabolize or how I process medications compared to another person with a different microbiome. And finally, all of these together have raised a lot of interest in how the microbiome can influence health and disease. And next slide, please. We can click through, please, with the data here. And so this is a really neat example of how you can study causal effects of the microbiome in the laboratory. And so as you can imagine, with humans, it's really hard to study. We can profile the microbiome, but it's really difficult to say whether any changes in our composition of bacteria or other microorganisms causes a certain effect versus whether it's just correlated. And so in the laboratory, you can use animal models to study this. And so this is actually a relatively early study in microbiome years. So back in 2006, Peter Turnbaugh, at the time from Jeffrey Gordon's lab, took microbiome samples. So essentially, these are stool samples from mice. And he took them from either genetically normal mice, so these ones on the right, and then a mouse line on the left that has a mutation in a hormone that influences when this animal feels full. So this is a hormone that's required for telling the brain that it's conferring a satiety signal, so telling the brain when this animal's full. And as you can see, if you delete this, then this is a mouse model for obesity. And so what he did is he took microbiome samples from these mice and transplanted them into normal mice, and found that just by transferring microbes alone, he could influence body weight. So essentially, the mouse that received microbes from this mouse model obesity started gaining more weight compared to controls. And subsequently, and in several other studies not shown here, labs have replicated this with humans. So in this particular study, identical twins were examined. One twin that was diagnosed with obesity, and the other twin that was lean. And taking stool samples, transplanting them into mice, and seeing that mice that received the obese microbiome started gaining more weight compared to controls. So these types of studies are really powerful because they can actually tell us or indicate causality versus correlation. And similar approaches have been used for many other symptoms, like metabolic disease-related symptoms, and more recently, even behavioral symptoms. So next slide, please. And so this leads me up to my area of research. So my lab is really interested in how the gut microbiome may impact the brain and behavior. And so there's a lot of exciting evidence suggesting that the microbiome can influence the brain, especially here depicted in the gut. So this idea that nutrition, of course, is very important for brain and behavior. And then also more recent findings that specific microbes can influence production of chemicals like neurotransmitters and others that could influence the brain in a more specific way. And next slide, please. And if you could just click through. And so overall, my lab is really interested in these very basic questions, like how do microbes communicate with the nervous system? Can we find out of these thousands of species, which ones have a particular effect that's specific to these particular species of bacteria versus others? And how do these interactions between microbes and the nervous system impact health and disease? And finally, this really neat question about what is the reason, if any, of an evolutionary benefit for microbes to impact brain and behavior. And so these are the really fundamental questions that we're really interested in studying. And so I think that's my last slide, so thanks.
So you guys alluded to in your introductions to the new scientific methods that are enabling you to address new questions about microbes, how they affect our health, microbial communities, and fermented foods. Can you comment on how the new developments, for example, in sequencing and imaging technologies are changing the questions that you ask? And I mean, Sandra, this question goes for you too, because you are working with Rachel and her team to be able to understand more about the microbial communities and how those affect the health of people that eat fermented foods.
Yeah, sure. So I the technologies have been really really spearheading, I think, the development of this area of research, especially in the microbiome stuff, because sequencing has become so inexpensive that you can use it, day to day in your experiments. So there are also crowdsourcing. I don't work for them, but there are crowdsourcing efforts where you can receive a kit for a really inexpensive amount of money, and then profile your own microbiome and get this data back in a relatively short amount of time. But at the same time that technologies have really driven this area of research, there's a long way to go. And so I think the wave of the future is really further advancements in technology. So for example, right now, a lot of approaches that are used to study complex microbial communities take a bulk sample, say a cheek swab, and you take this entire community of hundreds, if not thousands, of different types of bacteria, and you profile it all together. And this is just one sample and one cross-sectional time point. But I think a really huge advance or a need in terms of technology and development of technology in the area is being able to look at spatial structure. So instead of just getting, a complex sample and all of these bacteria are mixed together and disorganized, and then you get, data spit back in an Excel sheet, could you actually image these very complex spatial structures and see which bacteria are close to each other and which are far apart? How do they form complex communities? Is there a structure where there's a core of different types of bacteria and a perimeter of others? And so these types of investigations into spatial structure can tell us a lot about interactions that we don't learn very much right now from a bulk sample.
Yeah, maybe I'll follow up a little bit. So, and actually we're starting to do some of these spatial structures. You saw the picture of the sort of cross-section of the rind. You can just imagine all the interesting kind of spatial organization of microbes there. But I guess one thing is, in terms of how this technology has changed the field, it's completely transformed our view of the microbial world. And part of that is because, we talk about the fact that there's these hundreds or thousands of different species that we now know are in these different environments, but we couldn't actually describe what they were before without the sequencing technologies. And that's because when we try and bring these microbes into the lab, they're not very happy with us. And so they basically are completely stubborn and refuse to grow in the lab. And so it's been known for a long time that there's probably much more diversity in the environment than we actually can measure because we can't grow the organisms in the lab. And so what sequencing has allowed us to do is not rely on these microbes to grow in the lab, which they refuse to do in many cases. We can actually go out to any environment on the planet and take a small sample and extract all of the DNA from that environment and just sequence the DNA. And that within that DNA, every microbial cell in its genome has essentially what we can use as like a fingerprint. And so different species will have a different fingerprint, and so we can actually describe all the different species based on their genetic material. And so now we know that there are these hundreds or thousands of species, and we can actually give them names. And we can observe how they change over time or from place to place. But then once we do that, then we have to figure out how it all works. And that's, I think, where a lot of us are in the field right now is saying, okay, we now have described the patterns of diversity, but how does it actually all work? What drives the formation of these communities? And so that's, the sequencing technologies have completely changed our view of the microbial universe and expanded what we know in tons of ways. And there's a lot of labs that are pushing even further on these technologies and developing new technologies. And so it's a really exciting time to be a microbiologist right now because we're learning so much about how much diversity there is in the world and how important these microbes are. And now we get to figure out how it all works.
And I don't understand these technologies at all. And at the same time, I'm just constantly sort of like reviewing abstracts of the studies, and it's just really fascinating what all these investigations into microbial communities are revealing about the function of our bodies, about these different foods, about the soil, about the environments in which we exist. So I feel really sort of thrilled to get to sort of see this area of scientific investigation sort of filling out our knowledge. And all of us who were born in the 20th century were indoctrinated into this war on bacteria and this sort of thinking that like we should avoid bacteria. When we encounter bacteria, we should kill them. And hopefully this these realms of investigation demonstrating just how important bacteria are, is beginning to sort of shift that and at least give us a more, nuanced view of the microbial world. But, from my perspective of, functional fermentation, I would say that, microbiology and sort of, the earlier period of microbiology, which was all based on sort of, isolating organisms. And a lot of the traditional fermented foods have been basically simplified by this. And an example that I like to use is yogurt. Like how many people here have ever tried making yogurt? Okay, so maybe some of you have had the experience that I had. You bought plain yogurt at the store. You use that yogurt as a starter. If you get the temperature manipulation right, your first generation of yogurt is beautiful, thick, tangy. If you use that as a starter, your second generation, you're going to have it's never going to be quite as thick as the first generation. And then by the third or the fourth generation, it doesn't even have the characteristics of yogurt, so you have to go back to the store and buy a starter. So why is it that we can't perpetuate it? And how did this food ever, stand the test of time and get passed down through the generations if it's not reproducible? And it turns out that the reason is that the yogurt that's commercially available is a simplified version of traditional yogurt. And if you can, find an heirloom yogurt culture, you could keep it going. I mean, it's commercially available now. I mean, I bought one from culturesforhealth.com, and I've done, hundreds of generations with it, literally. But it's a more complex community, and microbiology always viewed these complex, unknown communities with some suspicion. Let's isolate the ones that are functionally necessary and get rid of the ones that we thought were extraneous. And so a lot of fermentation processes have been sort of simplified to pure cultures, which generalist practitioners, people making them at home, can't possibly, they rely upon cultures that have to be isolated and propagated in a laboratory. So I feel like while microbiology has illuminated these processes, it has also in certain ways disempowered the generalist practitioners who have, really kept these, cultural traditions, alive through the millennia.
So how is this next generation level of knowledge that's coming from some of these experiments, like your collaboration with Rachel, which you should share more about, but how is that changing the way that you're thinking about your functional fermentation and changing, adding value to what you do?
Well, I mean, for me, like the biggest thing is that in the popular imagination, we are moving from the idea that bacteria are our enemies to a recognition that bacteria are, a fundamental matrix of all life, and that, all forms of multicellular life incorporate bacteria and are dependent in many ways on bacteria. So, I mean, I would say that the biggest thing is, helping people realize that bacteria are just not a force for evil that we need to eradicate. And in fact, we could not possibly, survive or sort of, mediate our lives on this earth without, cooperation with bacteria. And I think, everything else, stems from, sort of that, basic recognition. And what I've been, the collaboration I've been working on with Rachel and others in her lab at UC San Diego, has basically been just swabbing lots of different, I mean, it's very recent, so we don't have any results, but I do these workshops. I just had a week-long workshop where I live in Tennessee, and, we made probably 30 different fermented foods and beverages over the course of the week. And we swabbed them all, at different stages of their fermentation. We weren't completely comprehensive, but we tried to get as many of the raw ingredients before we fermented them. And then the students, the first day when they arrived, we did samples of our bodies. The last day when they left, and then a week after they left. So it was basically to see whether a sort of intensive consumption of these kinds of foods had any appreciable impact on the individual microbiome. So I'm really looking forward to seeing what they find.
It'll be fascinating.
Yeah.
So maybe you can give everyone a sense of the timescales when you guys are studying the effects of gut microbiome and microbial communities. How quickly do they change? Like if you stop eating a certain food or something.
Right. So my lab was involved in a study that was published a couple years ago where we were able to show that quite dramatic shift in the diet. So, this is going from sort of a normal diet to maybe eating no carbs or to a vegan diet, for example. With that sort of dramatic shift in your diet, you can have a major impact on your gut microbiome within a day. So it's actually quite rapid. So in sort of designing the studies that we were doing with Sandor, we were able to anticipate that we wouldn't actually need to wait very long to see these microbes sort of going through your digestive tract and out the other end. And, and so we were able to kind of incorporate that into our timeline. But with the technologies as they are now, it can be very rapid, the analyses that you do. So Rob Knight, who's also part of this project with Sandor, is a professor at UC San Diego in the School of Medicine, just published a paper showing, a 48-hour turnaround from collecting the samples, sequencing the DNA, and also looking at the metabolites and analyzing the data. So it's actually, the technology is at a point now where we could very rapidly analyze samples and sort of get a really nice picture of the microbiology of any environment. And I think one of the things that I'm excited about in this project that we're doing with Sandor, so the design of the study is really to look at how diverse these traditional fermentation processes are. And it's interesting because these technologies are so recent and the traditional fermentations are so old, the two haven't really met up anytime in the recent past. And so, my lab was sort of one of the first to apply these technologies to these cheeses, and so now we're trying to apply them to some of these other traditional processes. And so I think it'll be interesting to see what these new technologies are able to tell us about the diversity of these very different types of fermented foods, and then what happens to your microbiome when you consume these foods.
And Elaine, did you want to comment at all on timescales involved in the effects of gut microbiome and your brain chemistry?
Yeah, sure. So similar, at least with these extreme diets that you were mentioning. So, for example, my lab is currently studying a very, I guess, extreme diet, which is essentially no carbohydrate, a very high fat, ketogenic diet. And so this diet has been historically used to actually treat pediatric epilepsy. So it's this rare case where a diet has been used for almost 100 years to treat seizures, and that it's this rare case where a diet has actually been integrated into clinical practice. So where hospitals and clinics around the world will enroll patients and use a diet as a therapy. And so in the laboratory, we could see that in animal animal models, if you put mice on this diet, you can see a really dramatic shift of the microbiome within a couple of days. And actually, our data, preliminary data at least, is suggesting that this is important for seizure-related outcomes, so the benefit of this diet. I should say that this has not been tested in humans at all, and it's really preliminary findings. But it's really suggesting that in really short time scales, you could see microbiome changes that could impact brain and behavior.
Fascinating. So I should solicit any questions. I don't know if there are question cards circulating around out there, but please, volunteers will collect them and bring them up to the front. So Sander, maybe you can comment a little bit more on this trend, this rise in this interest and obsession with fermented foods. And of course, this is due to your infectious enthusiasm, but what are your observations of why this is catching on, why kombucha is so popular?
Okay, so first of all, kombucha aside, I like to challenge a little bit the idea that like sort of suddenly fermented foods are so popular. Like if you think about fermented foods, encompassing bread, cheese, wine, beer, chocolate, coffee, I actually never noticed when these foods waned in popularity. So
well, and of course, we've been eating them for thousands and thousands of years.
So I think that most of the products of fermentation have had really enduring a popularity. And if you walk into a gourmet food store anywhere and look around and think about the nature of the foods that you're looking at and smelling and tasting, they're almost all products of fermentation. And fermentation just creates these, compelling flavors. I don't have encyclopedic knowledge, but I haven't been able to come up with any examples of culinary traditions that do not incorporate fermentation. But as to why there's growing awareness of it, growing interest in it, I would say that there are two broad reasons. The first is that there's growing interest in food. And, after a couple of generations of people, being thrilled to, just have, convenience food, one-stop shopping, prepared foods, we're sort of in a generation that is, sort of seeing some of the downsides of that. Convenience is great, but if the products of our food system are not nourishing us well, are causing all of these diet-related illnesses, then people are noticing, maybe there's a sort of diminished nutritional quality of some of this sort of food coming out of this mass-produced convenience food system, and maybe the technologies of growing food in this way are environmentally destructive, and we're making connections between environmental destruction and the methods we're using to prepare food, and maybe some of the sort of economic devastation has to do with the centralization of food production and the disappearance of agriculture from the landscapes in many places. And so people are wanting to become more connected to their food, and once you start interrogating your food and thinking about how it's produced and where it comes from, and fermentation is just part of the answer and what you start learning about. Then the other thing is that after a century of bacterial panic and terror about bacteria, we're beginning to recognize the importance of bacteria. So there's a sort of allure to kombucha and sauerkraut and kefir and yogurt and other bacterially rich foods that have probiotic qualities. And at a time when people are beginning to be aware of the microbiome and the bacteria inside of us and all the important roles that they play in our physiology and our well-being, and how diminished they are by antibiotic drugs and antibacterial cleansing products, and maybe even the chlorine in our water. People are seeking to restore biodiversity, and these foods are a great way of doing it. So those are the sort of two main, reasons that I see a growing interest in them. But the foods themselves have always been popular.
Mm-hmm. Yeah, and maybe I want to add a little bit to that, which is that I think the increased interest in food and people wanting more and more delicious and more interesting foods, the traditional fermentations, because they really rely on much more diverse sets of microorganisms, each of those different microbes has their own sort of flavor to them. So when we're eating these microbes, they all taste different. And so the traditional products have much more complex flavors. And I think this sort of goes back to what you were saying about the simplified yogurts and other things like cheeses that have been really reduced in terms of, let's just take this 1 or 2 species that we need and not really take into consideration the overall flavor of the food. And so I think by going back to some of these traditional fermentation processes, we're encouraging more diverse microbial communities, and that's giving us a much more complex set of flavors in the food, which makes it more delicious, usually.
And we've
Yeah.
also seen here in the Science and Food series over the years, if some of you attended the lectures by David Chang and Rene Redzepi, some of these high-end chefs are, I think, really embracing fermented foods because they can create such unique flavor profiles, and they're really sort of local, indigenous organisms as well. So one of my favorite stories is when, well, I think David Chang served some of his misos here and vinegars, and as he was getting into fermented foods, but he was very fascinated by understanding more about this fermented, well, it was really molded pork product that he was making to substitute for katsubushi in the dashi broth, the stock that they were making in the restaurant. So they buried this piece of meat into some rice, and one day when I was visiting the R&D lab in New York City at the time, they pulled this out and they said, "Look at this, what do you think?" And it was just this piece of rotting meat. And they said, "It tastes really delicious, but we don't know if it's safe." Which is when they shipped samples to Rachel, who sequenced them. But anyway, this just goes to show how there is this movement, I think, in restaurants as well, to the extent that there's R&D, research and development laboratories that people like chefs David Chang and Rene Redzepi are supporting as well. So were there some questions that we have from the audience here? Hannah? So speaking of health benefits, maybe you guys can speak to that. I know this is an intense question. I'm sure there will be more questions about this than here, but what is known? I mean, I realize this is still an open question to the extent, but types of health benefits from eating fermented foods, and what about probiotics? Like probiotic supplements.
I'm sure. I mean, I'll take a stab at it. I would say that any kind of bacterially rich food, a fermented food that hasn't been cooked after its fermentation, is probiotic. I mean, certainly people in the probiotics industry, the people selling little capsules, would argue with me on that point, because I mean, most of the probiotics that are for sale are proprietary strains that have significant research behind them, clinical trials, and really, like, nobody's doing clinical trials of sauerkraut. And actually, nobody stands to, no individual or company stands to gain from, constructing trials of sauerkraut, because sauerkraut's in the public domain. So the things that are being tested are these proprietary strains. But if we think about probiotics as or the objective of probiotic therapy as some sort of restoration or rebuilding of biodiversity, then certainly the traditional fermented foods and beverages embody much greater biodiversity than any probiotic capsule. My own perspective, the way I try to sort of talk about it to people, is that because of all the things we've been hearing about the roles of bacteria in our guts and in our bodies, and the important role that they play in our ability to digest food, metabolize nutrients, synthesize essential nutrients for us, regulating our immune function, what we're learning about how they regulate our brain chemistry, then it stands to reason that building biodiversity can potentially benefit any of those processes. But I think that, the potential benefits are really broad and general, and they're not really specific. It's not that I could say that there are compounds, isothiocyanates, that have been identified in sauerkraut that are regarded as anti-carcinogenic. Does that mean that sauerkraut is an effective treatment for cancer? I personally have my doubts. I think it's a great thing to eat along with whatever therapy you might be pursuing. But I think we have to think about these things in holistic ways, and we can't really expect that eating one individual food, drinking one individual beverage like kombucha is necessarily going to cure diseases. They're sort of part of the larger picture of our health, and the bacteria in us are a huge part of the of the picture of our health. But we don't have enough information yet to say that eating this particular food will rectify this particular health problem. And yet I hear anecdotal stories all the time of people who feel that their health has been substantially improved by incorporating these kinds of foods.
Yeah, I can add to this. So I think that the promise for kind of new generation of probiotics or microbe-based treatments is much greater than what's out there currently. And while there's, I agree, as was mentioned, that there's not that much known about health benefits of functional foods, even the existing probiotics, I would argue that there's not enough known about their actual health benefits. I think history has done the trial for us in showing that they're not harmful. That many of us take probiotics and eat yogurts, and it doesn't seem to cause any detrimental effects. But there isn't very strong evidence for an amazing health benefit either. I think maybe one area where there is some well-accepted evidence is that maybe for some GI complications like inflammatory bowel disease or irritable bowel syndrome, that current probiotics may be helpful. But what I'm really excited about is these recent studies into normal microbes, so normal microbes that we previously didn't consider, and that these normal microbes are already part of the human body, and just learning more about what they do usually in the human body to learn more about how to treat health and disease. And so these examples that I've showed about microbes that modulate the immune system and microbes that could modulate metabolism and even brain and behavior in the future, I think would be a redefinition of what it means to be a probiotic, whereas currently probiotic is a nutritional supplement or a functional food that you could get at the supermarket, but a future is live therapeutics, so bacteria or products from bacteria that have been rigorously tested in the lab, as much so as traditional pharmaceuticals, and that have clear indications for disease conditions. And so currently, you have to be wary of any products that you see out there that have very clear, probiotics that are available just over the counter that have a very clear medical indication, because in many cases, these have not been very well tested in the laboratory. But the promise, I think, is the most exciting part for the future.
Did you have anything else to add, Rachel? Or should we skip to the next?
Well, I think so I guess one thing I'll add is that a lot of the research on probiotics to date has really been focused on yogurt and a very limited number of strains. And I think there is some promise in some of the basic research that's been done in terms of thinking about how some of these microbes might interact with our immune system or interact with the microbes in our guts. And there's been some evidence suggesting that consumption of the microbes from yogurt, for example, can modulate the activity of the microbes that are normally resident in our gut. Although these microbes don't actually colonize our gut, and so it's sort of a case where you have to constantly consume them, and the actual impact of that modulation of activity is not really entirely clear if it has a benefit, a health benefit. And so I think, part of what I'm excited about is the fact that the focus has been very narrow on these yogurt species, but on a day-to-day basis, we consume millions of different microbes, whether it's the microbes that are on the surface of fruits and vegetables, or in fermented foods. And so I think there's a lot more diversity to explore in terms of what impact any of these microbes actually have on us, on our microbes or on our health directly, and whether that may be additionally some sort of next kind of generation of probiotics beyond sort of the limited range of species that we currently look at.
So could you lead us through like what happens if you take a bite of an apple, for example, or your cheese rind, and you ingest all these microbes and they go into our gut, which is very acidic.
Yes.
And so maybe some survive, some don't. Can you just lead us through how you guys are envisioning how microbes could be impacting our health?
Yeah, I mean, it's rough in there. It's not a pleasant environment inside your gut. And so, yeah, say you consume these.
I mean, for some of them, it probably is.
Yeah.
Right.
And many of the microbes that are in fermented foods, they're not well adapted to live inside of our body. They're used to living, say, in a dairy product or on the surface of a plant or something like that. And so we consume them, they go into our stomach, which is very acidic and probably kills off many of the species. And then they go into the intestinal tract, which is anaerobic, and there's, billions and billions of other microbes already there that are colonizing our digestive tract. And I don't think we really know what happens, as they go through the digestive tract. From some of the work that we've done and other labs have done as well, some of these microbes from fermented foods actually can survive the gastrointestinal tract. So we've been actually able to retrieve microbes from fecal samples of humans that have eaten fermented foods, so cheeses and salamis and things like that. So they have the potential to survive. Whether they're actually doing anything while they're there is something that we're interested in looking at now. That's sort of my sort of envisioning of what the microbes is. So it's not a pleasant environment for them, and I don't think most of them, we know they don't colonize your gastrointestinal tract, at least these ones from food.
I mean, what I would add to that as the non-scientist who has been sort of, skimming through lots of papers, is that there is an interaction, and we're trying to figure out, like, what does that interaction consist of? And, to me, as a sort of, non-microbiologist, one of the most fascinating aspects of bacteria are their genetic flexibility and all of these sort of mechanisms that bacteria have to, exchange genetic information, pick up genetic information from the environment, discard redundant or unnecessary genetic information. So, really what I'm envisioning is genetic exchange happening during this, passing through the intestines, and that the bacteria in the food that we eat are enriching the genetic environment in our intestines and giving the bacteria which have colonized the intestinal tract, just more information to work with.
And that's actually been shown to be true.
Okay.
So there's this really wonderful study published a couple years ago looking at the human gut microbiome of different populations, and they were examining the genetic material of the gut microbiome from a Japanese cohort. And they found some weird genes in the microbiome of this population, and one of these genes was made the machinery that was required to degrade seaweed. And so normally our microbiomes don't have the ability to degrade the complex polysaccharides that are present in seaweed, but there are microbes that normally grow on seaweed that can degrade these carbohydrates. And so what appears to have happened is that in the Japanese population, the consumption of seaweed and the microbes associated with seaweed, the genetic material from those seaweed-associated microbes jumped into the gut microbiome of the Japanese population, and probably because they're eating seaweed, those microbes had an advantage because they were now able to help degrade and acquire more nutrients from seaweed. So definitely we have already seen examples of this, and there's probably many more to be discovered.
Genetic modification in the wild.
Yes.
Yeah.
And Elaine, you were going to add something?
Oh, yeah, so I just wanted to add to this idea that even though you can have, typically I think when you ingest microbes in food or in probiotics, there isn't really evidence that these microbes actually stick or colonize. But that doesn't mean that, that they can't have an effect. And so there are studies on yogurt microbes, for example, and just showing that even though they are just passerbys and you can retrieve them later from stool samples in humans, that even in passing by, there could be transient interactions with our own microbiomes. And you could see that, that the responses in our own microbes are changed even with ingestion of these foodborne ones. And so even if they're passing by, they could potentially have an effect on our own microbes.
And talking about feces, I mean, fecal transplants, though, that's very well established to have an effect on your health. So,
yeah, so probably Elaine knows more than
an
I do,
effects
but-
on obesity, at least in mouse
Yeah,
models,
so
but-
there's a lot of recent work on fecal transplants in the medical field, and their utility in the treatment of Clostridium difficile infections. So this is an infection that people will often get if they're undergoing sort of surgeries or have other major issues where they're hospitalized and given lots of antibiotics. And the idea is that you are disrupting the ecosystem in your gut, and Clostridium is sort of there maybe normally in low populations, but once you wipe out the rest of your microbes, it can proliferate and colonize and cause really terrible infections that are recurrent and difficult to treat with antibiotics. And this method of taking stool samples from healthy individuals and reintroducing it into the colon of people with this disease is incredibly effective. And they actually recently were doing clinical trials on this and had to cancel the clinical trial because it was unethical for the control group that wasn't receiving the fecal transplants. It was so effective in the clinic. So it's a really exciting sort of advance, and it's something that I think that's been around for a while, but it's now just being more and more appreciated and studied in terms of its effectiveness.
So, Elaine, any evidence of fecal transplants impacting brain and behavior?
Too early to say. I think definitely in all of these. So currently, you can look at disease populations. You know, you can pick any disease, and in many cases, you can find some data suggesting that there's a change in the microbiome. But the big mystery there is whether this change in the microbiome is just a side effect of the disease, or if it's another side effect or a consequence of medications that these individuals are taking, or dietary changes or lifestyle changes that they've had, or whether these microbiome changes actually contribute to the disease, like in C. difficile infection. And so there's been a lot of interest in applying fecal transplants to other diseases aside from C. difficile infection, but still it's early days, and there aren't really clear conclusions from that.
Well, it's certainly very exciting that it is in the early stages of more discoveries, and along those lines, here's a question on, similar to the discovery of new planets in our Milky Way, are we discovering new species of bacteria in our bodies and our food? how long have they been, how long have they been known to us? And then just an addendum to that question is how you go about naming them. Like, do you name them like new planets? Or you mentioned, Rachel, earlier that that there was the renaming of some existing bacterium as well. So maybe you guys can comment on that.
Yeah, so there is definitely the potential to discover new species every time you look at a new environment. And because of the sequencing technologies, we're able to explore a lot more than we were before. So we haven't discovered any new species in cheese, but I bet if we looked a little bit harder, we probably could. You know, part of the problem is there's so many sequences you get back, so it's often difficult to even classify all of the things you have in an individual sample. But if you do discover a new species, then you have to figure out what to name it, and so name it after yourself, or somebody more important than yourself, or but yeah, it's exciting because every time we go out to a new environment, it's possible that we'll discover new species.
I just want to throw out as a non-scientist again that some of the microbiologists who I've talked to really reject the notion of species. And they feel like because bacteria have such genetic flexibility and are such shape shifters that it's sort of applying a concept that makes sense with animals and plants. To sort of something that's sort of much more highly variable. We have fixed genetics for better and for worse, but bacteria can become different kinds of things.
Yeah, so there's a classic study, well, sort of classic now, but it's not that long ago, where researchers sequenced the genomes of 4 bacteria of the same species, but isolated from different environments. So these are actually E. coli. Which is one of the best understood bacteria on the planet because so much research is focused on it. And so they took 4 isolates of the same species, and less than half of their genome was the same. And so even if we called something a new species, or maybe it's even the same species as we've seen many other times before, that doesn't really mean that we actually understand what it's doing or how it's doing what it's doing. So there's just, even beyond the idea that we could be discovering new species, the genetic diversity and potential that these microbes have is incredibly vast. So yeah, I think that's a great point, that these microbes are swapping genes all the time, picking up genes from different environments, evolving very rapidly. And so it's kind of amazing. Complex world out there.
And mind-boggling to think about how you would go about characterizing all these rapidly changing populations.
Tell me about it.
Yeah.
Well, which is why maybe it's more interesting to look at communities and like genetic range within a community rather than isolating the individuals.
Yeah, so we're actually doing a study in my lab right now where we're sequencing the genomes of species isolated from the same community, and we're seeing evidence of a lot of genetic exchange within this community on cheese. And so it's, and I think, the other, sort of going back to technology, we're now not just looking at species, we're sequencing all the DNA. So all the genes that are present in an environment, which gives us an idea of the total functional potential of any ecosystem, any microbial ecosystem. So we're getting lots and lots of more information every day. And so it's sort of one of the biggest challenges is figuring out how to decipher what this information is trying to tell us. Yeah.
So Elaine, what are some of the other big challenges for you and your lab and research moving forward, and questions that keep you up at night?
Yeah, well, there's some really, I guess, basic challenges. I mean, so currently, the types of questions that I'm interested in are really long-distance questions, so involving many organs. So I primarily study the gut microbiome and then interactions with the brain. And as you can imagine, there's this very long distance physically, and there's a lot that can happen from the gut to brain. And so one challenge is just is throughput. So currently we use live animal models to be able to study these long-distance questions, but this kind of limits us in how quickly we could get to answers. So how many out of these thousands of bacteria, if we went through one by one, it would take lifetimes to learn about functions for all of them. And so I think a really exciting challenge is to kind of develop more high throughput methods to learn about functions. So aside from being able to sequence and get names and relative percentages, can we develop technologies that allow us to learn about what they do just as quickly as we can get their names and their relative percentages. So that's something that I'm really interested in for the future.
And so coming back again to timescales and longer range, there's a question about what work is being done looking into the lasting effects of fermented foods on the microbiome. You guys were talking about how quickly the foods can have an effect on your microbiome, but what about lasting effects?
Yeah, so in the study that we're looking at now and in work that we've done in the past, we've kind of looked at after a couple weeks, and you can see the microbes kind of tapering off. So the microbes that you're consuming as part of the fermented foods don't really hang around for more than a few days or so, it seems like. So in terms of our ability to detect the organisms, that seems to be kind of within a week, I would say, would be my guess. We'll see what this next study tells us about a more diverse set of fermented foods. Whether their impacts on the microbiome last beyond that, we don't know at the moment.
Well, we're about to wrap up for the evening, but I wanted to give you guys the opportunity if there's any questions you have for the other panelists that you didn't get to talk about yet. And barring that, I think it would also be fascinating to hear more about the most interesting or disgusting fermented foods.
That sounds good too.
You can take your pick.
That's a good one for Sandor. Yeah.
I really haven't found any that I'm disgusted by.
Surströmming, the Swedish fermented.
Surströmming, sure. I've tried. Surströmming is growing on me. I've had it 3 times now. And,
this is the fish that's put in a can, buried in the earth.
It's this Swedish style of low salt fermented herring. And in contemporary production, they put it in a can, and it's ripe when the can bulges. I first tasted it at a conference in England, the Oxford Symposium on Food and Cookery, and they sort of made this sort of Swedish academic who was delivering a paper about surströmming and giving everybody tastes. They made her move it outside. And then there was a long line, and as I got close to the front and I could smell it, I was like, "Oh, am I going to be able to eat this?" Because it has one of these sort of sulfury smells that maybe is reminiscent of rotting food. But once I taste it, it's not like you're eating a big slab of fish. It's like a little piece of this very strong flavored fish, along with some sour cream and some onion, and a little sandwich on a cracker, and it was really delicious and left a great aftertaste. And I went and had another taste. But I'm a food adventurer. I love to taste different things, and I love to make different things. And I haven't really found anything that's just conceptually. I've made there's a style of chicha. Which is a sort of like a South American corn beer. And in the mountains in South America, one of the ways of doing it, probably the most ancient way of doing it, is people chew the corn, and enzymes from saliva amylase enzymes break down the complex carbohydrates into simple sugars fermentable into alcohol. And I have done this with students with corn, with cassava, and with potatoes. And it's really fun and interesting just to see people's visceral responses to the idea of drinking something that other people have chewed and spat out, even though, you are boiling it, you are brewing it for hours in between the, sort of the chewing part and the drinking part. People. Fermented foods are controversial. They're flavors. Stinky cheeses, right? As my taste has evolved, like if I could smell a cheese from the back of the auditorium, I'd be sort of distracted this whole time. And I'd be like, when can I have a taste of that? And I'm sure at least a few of you would walk in here and say, did something die in here? And you would never for a minute think about putting that into your mouth. So, one of the reasons people love fermented foods is because they have strong flavors, but, they're also, they're acquired tastes, and people who haven't been exposed to them often find kimchi or sauerkraut or strong cheeses or surströmming or any number of foods to be disgusting. Even though I think they're delicious.
So speaking of which, there are some samples. Are people eating them? Do you have them? Right. So you have some samples of some kimchi and cheeses you can think of as you eat into the rind, and kombucha as well. So there's so many other fascinating questions. I mean, we could go on for hours. Microbiome in a gestating infant, love and microbes, how stress affects your microbes, the best diet to increase your microbes. So unfortunately, we're out of time for this evening, but do stay tuned on our Science and Food blog. We will be making attempts to answer these questions over time, but I think we've had a fascinating evening, and some of the take-home messages include microbes can be good to eat, and they're not pathogenic. They don't always cause diseases, and we're excited to have brilliant scientists like Rachel and Elaine who are forging the way into studying these complex systems and addressing questions of how microbes affect our health and our brains and how we can understand communities in the laboratory. And we're looking forward to seeing the outcome of your collaborative research too with Sandor. So on that last note, there's a final slide. I just wanted to let you know there's one last event in our Science and Foods 2016 series next week, next Thursday, right here, same place, same time, with guest speakers, climate scientist Peter Fried, chef Anthony Mayen, and Chris Ying of Lucky Peach, who will be talking about carbon footprint in dining. So we also have some surveys. We would love your feedback. Laura is carrying them right here, and we're offering free tickets, a set of 4 free tickets to next week's event that you will have a chance to win if you complete a survey. So help us out. This helps us to guide the planning and organization of future events and address any desires you may have for other events on food you might like to see here at UCLA. So on that note, thank you all for coming, and thanks to our speakers.